Hello everyone. Thanks for attending this talk. Well, when I read for the very first time about this symposium through Twitter by Leighton Wright, I thought it was a good opportunity to show these results, but mainly to tell a little bit about the context in which they were obtained. The experiment was set up as part of an offset coming from the construction of this dam, Hidrosogamoso, at the northeast of Colombia. The idea of seizing a standard restoration project to do some research came to my mind due to several circumstances. First, an initial step to restart my professional career in my country after completing the field stage of my PhD under the supervision of Richard Hobbs. Second, the very particular conditions in Colombia to do restoration, in which implementation is in general well funded, but wherein the lack of a research culture makes those resources are wasted at a worrisome frequency. This same lack of research culture has caused the emergence of publications like the one you see in the slide. Although it encompasses interesting research hypotheses, it has been titled and perceived as a prescription, which unfortunately ignores the omnipresent contingency inherent to ecosystems dynamics. The design of the intervention in all fields of our study area was prompted by the early work by Saxahawi, Karen Hall, Leighton Wright, among others. As a necessary clarification for those new in the topic, it is important to say that the most recognizable type of applied nucleation at restoring are islands made of planted trees. So this special pattern of plantation replicates natural patterns of regeneration and repeatedly has been reported as a cost-effective restoration strategy. So once we designed and implemented the experiment between 2015 and 2016, we realized that the idea of doing research through projects had been previously posed, at least in this paper from 2014. And recently in 2021, through this work led by Sarah Wilson, we also realized that our idea of finding optimal species assemblages at applying nucleation was among research priorities. The study area has mesic and stable abiotic conditions throughout the year that probably gives biotic interactions the major role at directing successional trajectories. Moreover, the area had a relatively brief land use history that, along with abiotic conditions, give chances for quick recovery, but also to quickly test research hypothesis. So the experiment was set up in two different sites, each with an oil-field area equivalent to 10 hectares, and in this scheme you can see the spacing between nuclei in each of these oil fields. In this other drawing you can see both the design of the plot that we use, containing the subplots to monitor the frequency of invasive grasses, and also you can see the spacing of trees within a single nucleus. Thick dots correspond to trees of locally threatened species, and dots without the areole correspond to PIONNER species whose assemblages we tested in the experiment. The list below describes the composition for each treatment. Treatment 1 and 3 are species typical at a secondary forest, forest gaps, and forest edges. Treatment 4 is made exclusively of treelets that commonly colonize oilfields. Treatment 2 is the combination of all the previous ones. Okay, talking about results. Around 2 years after plantation, in general, vegetation structure and diversity achieved values similar to these found in reference forests. You can notice that I did not include Urera and Solanum treelets at nucleate type 1, as most of them died, and by the time of monitoring, almost all contribution to a structure came from the three species Heliocarpus americanum. It's important to say that these data correspond not only to survival planted species but also to the vegetation that spontaneously appeared after plantation. Only in one case, values kept the same between control plots and nuclei. It is for stem density at comparing nucleate type 2 with controls. This similarity is due to high presence of the tall grass Megathyrsus maximum at those plots, and density was estimated from diameter at breast height equal to 1 centimeter. Before presenting the second result about the abundance of graminoids, I will tell you about the situation in that regard before planting. So, there were patches of two different species with records of invasions in other parts of the tropics, namely Brachiaria radicans and the tall grass Megathyrsus maximus. You can see why I'm talking about tall grass. Well, the small blue stain that you see in the picture of this grass is one of our research assistants. You can see the situation was a bit scary in terms of overcoming such filter at the beginning. To do so, we clear grasses before planting, but we have to do it twice more monthly after planting. Now let's see the results. Remember that abundance of graminoids was recorded using a grid of 60 subplots inside the 20 by 5 meters big plot to measure tall vegetation at each tree island. Although nucleate type 2 and type 4 did not have an abundance as higher as these of control plots, they had a higher grass abundance compared to those of nuclei dominated by trees like Heliocarpus and nitrogen fixers Inga and Erythrina poepiciana. Talking about survival of locally threatened species, I have to tell you that these threatened species were selected from interviews, conversations, and surveys that we made with sawyers in the study area. Also, you can see that most of these species are late successional Lauraceae. So, as with abundance of graminoids, the best nuclei at promoting establishment of these species were these formed either by Heliocarpus or by nitrogen fixing tree species. Well, linking final thoughts with what motivated this talk, we can say that the restoration project that gave origin to this experiment was successful. In 15 out of 20 cases, coming from 5 variables crossed with 4 treatments, structure and diversity of vegetation reached values similar to those found in reference forest. This would be the result of landscape context, land history, and the fact we control graminoids several times after planting. On the other hand, the more diverse assemblage was not the best, and hence, as far as we are aware, this is in opposition to vast evidence in the literature. Thus, we will get deep into it to explain this trend we found. Finally, and without having reviewed the literature rigorously, we wonder if size when adult is a good predictor of success at selecting early and mid-successional species at restoring degraded forests heavily invaded by gramineads. If trees, but not treelets, worked well for us in this study, we also wonder if they would work equally well at harsher environmental conditions. So, thanks a lot for attending this talk, and also great appreciation for all these people that make possible this restoration project that turned it into a research project. Thanks a lot.